Hello guys. Now today we will start with our remaining topics. We'll start with the cell lines. So cell lines basically means, suppose take an example. This is an tissue. So if you take a tissue means what? Tissue is the complexed organ, or it is the complex system of organ. And suppose that tissue is embedded with multiple or different types of cells which is present and performing a single function. So if we extract some part of the tissue, we can find that there are different types of tissue, like red one is different, the blue one is different, then this one is different. So like there are different types of tissues are there. And if if we culture them primarily, then you can see that we will find out that we will get fibroblast cells, then keratinocyte cells, right, then melanocyte cells. So different types of cells we generally get out of these heterogeneous cellular population. So next part that we want to see that what is the cell line. Basically, if we want to define the cell lines, how we can define cell line. So here the definition is, once a primary culture, okay, once the primary culture, now here the primary culture is already prepared, or we have made a primary culture in our laboratory first of all from the crude sample of tissue. And once we subculture it, now earlier in my lecture I told you that once the primary culture is we make in the flask, what do? We, once the confluency is there, okay, once the confluency is there, the confluency means what? There is a like take example, this is the flask, I want to draw it, this is the flask, and whatever heterogeneous population of tissue is there, it is generally it's sprayed all over the flask, right? The confluency is there, confluency. Now this confluent culture means what? This no more space which is allowed to the cells to grow or to regrow. And so at that time what we need to do, we need to take that culture and we need to subculture them. Okay, so once we subculture it, or other means is the, it is the passage, it becomes known as cell line. So that is the cell line. This term implies the presence of several cell lineages of either similar or distinct phenotypes. So like take example, this implies what? The presence of several cell lineages. Now what is meant by cell lineages? Now there is other term, cell lineages. Now what happened? Take example, single primordial cell is there, right? Single primordial cell is there, and that single primordial cells, now we are the cells is now dividing, right? It is dividing in 2 or 3 or 4, right? The 2 cells form, then after that the 4 cells form, right? Then 8 cells form. So whatever cells form, suppose we are looking towards 64th generation cells, okay, we are now looking towards 64th generation cells. And what is the characteristic of these cells? Might be it is different from this primordial cell, or might be it is different from this second cell division which is present over here, this cell. So it has some another quality or another property, but from where exactly this has differentiated? These primordial cells. So whatever cells which is comes after cell divisions, whatever cells which is present up to the 64th generation, these are all cells, we can say that they are from same cell lineage, so they share same lineage, right? That's what we called it as lineage. I hope so you have cleared your concept over here. Then take example, like that's why here it is written either the similar phenotype, phenotype means character, the representative character, phenotypic character. like the cell surface is having a kind of different cell receptors. Might be they are having more lipids, more receptor channels, which is made up of glycoproteins. So like same character or distinct characters or different characters, and that's what we call as phenotypic characters. If one cell lineage is selected by cloning or by physical cell separation or by any other selection technique to have certain specific property and that have been identified in the bulk of the cells in the culture, and this cell line becomes known as the cell strain, right? I hope so you have understood this topic: cell line, cell strain, and cell lineage. So here there is a table, there are few tables like which states that commonly used cell line. So commonly used cell line, why cell line is very important, guys? This is very important thing. Uh, what I want to say is that this is important topic now. A common cell line, what is meant by common cell line? The common cell line, that means suppose there are two types of cell lines that we have already seen in our lecture, like one is finite cell line, the other one is infinite or continuous cell line, right? I hope so you have understood these two concepts. Now here the finite cell line, IMR 19, MRC 5, then MRC 9, right, then WI. So these are basically cell lines which is derived from a particular organ. So what is the organ of which is the cells which is originated from? That is lungs. All the cells lines are belongs to lungs, and they are all morphologically fibroblastic in nature. So they are fibroblast cell lines, right? And they belongs to human species. Now here I want to tell you that if the cells belongs to humans, then of course you make consideration that these cell lines could be useful in clinical trials or drug testing, antibiotic testing, anything which is used further in human body or in human medical sciences. So at that time these cell lines are very much important. Now from where exactly they have originated or taken? They are embryonic, all are from embryonic stem cells or embryonic. Embryo is the basic origin. And then what is the ploidy condition? All are diploid in condition. The characteristic here, it is written, it is susceptible to human viral infection, okay, or contact inhibited, susceptible to human viral infection, susceptible to human viral infection, contact so these all are susceptible to viral infection. So that means what? Whatever virus, take example coronavirus. The coronavirus infection is merely basically to which cells? I hope you got my answer. It is the lungs, basically, you know that. And so in laboratory might be there are chance, might be there are chances, they were using these cell lines, which is very popular cell lines, and they were observing what is the side effect, what is the effect, what happens to the cells, what they secrete, what kind of cellular factor or markers that they represent on the cells after infection. And so based on that they generally observe the post infectious effect of any viral disease on the body of human based on these cell lines, right? So that's why it is very important. And here there are several references from which reference or from which research this cell line comes in a science or comes in a study, right? Then the other cell lines are there, like continuous one. There are different 33, then BHK, then C7, HeLa, these different or Vero cell lines. These all cell lines are there, and these cell lines are basically continuous cell lines or infinite cell lines. And these infinite cell lines are basically what? These infinite cell lines, they produce as much as they can. So there is a no stop, or though there is a no any definitive pause or definitive stop to that cell line. And this is very important thing right here. So basically what I want to tell you that where exactly this continuous cell line useful. So where exactly it is useful? There where we generally working on cancerous cell lines, or some cell lines we need to study up to their extinct level. What will happen to the gene line, what will happen to the ploidy condition of these cell lines. Like you can say that the chromosomes ploidy or ploidy condition is aneuploidy, some are diploid, some are triploid, and sometimes the chromosome number multiplies and it goes beyond our imaginations after few subculturing of these cell lines. That is also wonderful, and it is the part of study to understand what are continuous cell line. Now here we are looking towards the culture age. What is the age, and what happens at the uh, to the cell lines if they are finite one or if they are continuous one? The cell lines with a limited culture lifespan are known as finite cell lines and behave in a fairly reproducible fashion, right? Like a fairly reproducible fashion. That means what? They barely divide and subdivide up to the a finite lifespan, right? There is a cell lines, and suppose they form a two cell lines, then they form a four cell line, then eight cell lines, right? So after that, what happen? Up to certain level only they can divide, and then ultimately you all know that the programmed cell death, apoptosis, this system ultimately initiates the self-killing mechanism or self-suicidal mechanism, and ultimately cell will die, and that's what we call as finite cell lines. And they grow through limited number of cell generation, usually between 20 to 80 cell population doubling times before the extinctions, right? This is the common or average cell population rate, right? Doubling rate. If we talk in terms of continuous cell line against these finite cell lines, they generally escaped from senescence control or apoptic control, right? They have escaped from that control, so the generation number becomes less important, and the number of passages since the last thought, like last they came up on the table in the laboratory, okay, from the refrigeration procedure, and like the generation number is unlimited. Okay, I'm here talking about the unlimited, continuously unlimited. That doesn't mean that they really grow unlimitedly. Some continuous cell lines have their own lifespan. Might be they get vulnerable, or they get die because of extensive amount of nurturing or extensive amount of nutritional feeding, excessive amount of CO2 inhalation or exhalation, excessive amount of hydrogen ion generation in their mitochondrial organ. So like different types of self-killing mechanisms are there, but not senescence, right? Not senescence. Because of any other factor they could die. Cell line designations, how we designate cell lines, right? This is very much important. Generally we observe, right? We saw in our that slide there is a term MRC5, WI38, or 3T3L1. What are they basically? What are they? They are like new cell line should be given a code, right? A code or designation. Example, like there is a cell line, there is a organ like normal human brain. So this is the organ, and so we pick up their initials. If we have prepared the cell line from brain, then there is an initial like NHB, normal human brain. Okay, the cell strain or cell line number. What is the cell strain, or what is the cell line number, or that lineage number? If several cells lines were derived from the same source, then like we generally put their names as NHB1, NHB2, NHB3. Okay, from the same source, from the same organ. Source means organ, from the same organ, but the different time that we are preparing different different batches of that cell line. If we clone that particular cell like NHB1, we are cloning again, then the clone number should be given after dash, right? The NHB1 or NHB2 1, NHB2 2, right? They both are clone, like NHB2 cell line is there, but they are different with the cloning procedure. I hope so you have understood the designation, or how we generally tag the cell lines on design the name. Now, why and how we choose cell lines? Suppose we want to work on some sort of drug right now. Take example, current example that I will provide. Bharat Biotech is providing a Covaxin vaccine to the humans or to Indian peoples generally for the vaccination against the coronavirus disease. Might be they have chosen in their primary study some cell lines. Now, why they have choose cell lines? There are certain characteristic or certain requirements. Like first of all, the cell line is finite or continuous. That we need to check. Now take example, if you are injecting vaccine to a human body, then of course our human body is contains all the cells which are finite, means they have a limited lifespan. We don't have any cancerous tissue or cancer in our body, so that we don't have any continuous cell line, so we have a finite cell lines here. Then next part is normal or transform. Now you might get confused with the finite continuous and normal transform. Normal and transform means what? Some cells are normal. They perform all the functions, metabolic activity normally. And some cells are transformed cells. So transformed one means what? Transformed one means the cells is the same. It is not the different condition, but might be it has changed genetic material suddenly because of environmental effect, and it has changed its expression. Okay, change its expression of what? Expression of protein. Might be in some proteins which is embedded, or some are inside the protein, or some are secreted protein. Might be they are different, and it is cells is showing something different phenotypic character suddenly because of environmental impact or chemical impact, any impact. And that is what we called it as transform cell lines. So that impact might cause that cell lines to become a continuous, or might cause that cell lines become a finite only, but a different one. I hope so you have understood normal and transformed one. So it is the important whether the line is malignantly transformed or not, that means cancerous or not. So there are different cell lines, like take example continuous cell lines. We saw example 3T3, BKK21, C13. These are batch number C13 or cloning number, and then this is the number of cell lines which is taken from that breast cell line, right? Next thing that we need to study, that is what is the species. Again we'll take example of our covaxin. Now covaxin is for human, right? So we need to take human cell lines which is acting against it, or which shows some sort of side effects or not any side effects. That's why we want to check that vaccine in the trial, and we need to take human species. After that, if you want to take a species which mimics the human, then they were used some monkey, right? Rhesus monkeys they have used, some 14-18 rhesus monkeys they have checked, and covaxin, right? I saw the research article, so that's why I'm telling you. I hope so you also know that. Now the species should be the same or belongs to the same category, like mammals. Why we have chosen rhesus monkey? It is more likely our immune system, our genetic complexity, and everything belongs like as the same as rhesus monkeys has or chimpanzees has. Or even though we use mouse also, because it is also mammal, right? But we cannot choose, take example any other species, right? We cannot choose honeybee. It is from arthropod family, right? Lepidoptera genus. So like we cannot choose that species for human antibiotic trial. So species is again very much important. The next one is growth kinetics. What is the growth? Suppose continuous cell is there. If suppose continuous cell, it is enormously grow, it is infinite cell line. But the cell, if the cell is belongs to some sort of a normal cell line, then it has some final, right? It has some final um, finite cell division number, where the continuous cell line don't show any finite one, it continuously grow. So that kind of growth kinetics we should know. The other property of growth kinetics is doubling time. What is doubling time? Then doubling time means the name is just sufficient, right? Name is just sufficient. The cell becomes 1 to 2, but in what time, right? But in what time they belongs to the same. At what time again the second cells become two cells and the other one become the other cells. So it these all structure, it's again depending on what, depending on what type or what kind of doubling time the cell has. Suppose the cell contains doubling time, like within a 2 days, right? Within a 2 days it shows some character within a 2 days. How could we do some sort of experiment? Can you imagine if you want to inject some antibiotic, or we are going to insert antibiotic in the cell plate or the culture plate, and the culture plate is dividing each cells one day after, right? Then we need a year to observe their post antibiotic effect on the cell line. So that's why doubling time is very much important. The another one is saturation density. What is the saturation? Saturation density means what? Whatever, how much saturation happens in the flask, how much saturation happens in the flask in how much time, and how much dense the population is, and depending on that, nutrient condition, antibiotic exposure, media exposure, this everything depends. Then plating efficiency. Plating efficiency means how much cells we need to plate a single cultural plate, how much cells we required, and based on that, how much efficient. Suppose that we want to enter 5 into 10 to 4 cells per plate, our culture plate, and that will show confluency within a half day or within a day. So that is not good plating efficiency. At least we need to observe this flask up to 2 to 4 days, right? So for that purpose we need to calculate our seeding culture, and we need to see their plating efficiency, right? That is plating efficiency. Then we need to check out growth fraction, then ability to grow in a suspension culture. These all the growth studies or growth factors we need to observe. Next thing we need to check out, that is availability of that cell line. If you are working on, again I'll say some sort of drug, take example penicillin, you are working on, you want to check post infectious effect of any bacteria, and you want to check how penicillin acts against that infection, acts against that bacteria or any fungus or mold, right? Then what? Then you need to see that if you want to work on that drug on the animal, and you want to see that you want to work on epithelial cell line, and you are selecting some cell line which is just available in some single Japanese laboratory, and you are working in New Zealand or you are working in India. Will you get that cell lines immediately within a week or within a 5 days? No, you need to wait for a month, so that the laboratory can approve their own cell line, so that their government can approve whether we want to send that cell line or not, and then our government should approve. Like, the availability in our country, availability in our state, and then in our city, it should be available, and how much time it requires up to the repositories make available to you. So that all the things are dependent. The next topic is validation. Validation means how well characterized is the cell line is, if it exists already or not, or can you do the necessary characterization. So these all are things we call it as validation, and we are going to see in our next lecture what is meant by cell characterization. So just wait for that. In our next lecture we will see characterization. But these all studies comes in validation. That means whether, like, take example, you are purchasing some sort of product, right? Take example stapler that we generally use in our academics to staple our uh, bunch of pages. If you are purchasing that, you don't purchase any false atom or first copy or second copy or some sort of Chinese material, right? You want some sort of hallmark on that, like ISO certification or any other certifications which is government generally validate that product is well for use and good for its own material, and that's what mean by validity or validation, not validity, sorry, validation, validated. The next thing is that phenotypic expression. What kind of phenotypic expression it expresses? Like, it expresses some hormone, the cell line, it expresses lipid moieties, it expresses proteins, or it expresses some other type of receptor material, or it expresses just an acidic material. That all expression things that you should know. So based on that you can choose your nutrition media. Generally when these repositories provide some cell lines to you, at that time they make sure that they should give each and every details of that cell line to user, Why? Because user is not going to work on that cell line again, because user's experiment is completely different. He wants cell line ready for its own practical, and he should have all the information about that cell line in his hand during the experimentation. So this phenotypic expression should be provided by the subculture, or sorry, repositories, and they made everything ready for them, users. The control cell line, again, that is the big thing actually. But control cell lines, if you are using the mutant one, transferred one, or you are experimenting on some cell line, so the same cell line should be ready at the time of experimentation as a control cell line, because whatever effect happens in the cell line which is in the experimentation, you can compare that with the normal one only, or control one, right? And then only you can decide whether there is a changes or not, or whether there is any type of change in gene expression or any other type of expression or not. Then the stability of that cell line. Stability matters, how stable that cell line is. Take example, HeLa cell line is not stable at all. HeLa cell line is very very robust cell line, and it grows like boom. It grows at like in very high speed, and there are reports in the laboratory from very good laboratories that HeLa cell lines make contamination in other cell lines also. HeLa cell line, because of it is a cancerous cell line. You all know that it's a continuous cell line, and it grows very faster, very robustly. So the cell line should be a stable, and has been cloned or not. If not, can you clone it? If you don't want to clone it, then don't clone it. Why to clone it? Then how long would this cloning process takes to generate sufficient frozen and usable stock for users only? I'm not talking about the repositories. Once you get the cell lines, you can multiply it and you can restore in your laboratory, right, up to your experimentation. You don't have to purchase again and again. So at that time you need to clone them. And what is the best procedure for cloning? Subculturing. You can subculture the fresh stock and you can preserve them in your liquid nitrogen facility, right, the cryopreservation facility. So that thing we can do. The next thing which we need to study is routine maintenance. What is routine maintenance? You need to observe culture. Once your culture is initiated, then whether there is a primary culture or subculture, you need to study. If it is a subculture, then you need to change the medium periodically, right? Sorry, you need to change the medium periodically. Then what is the feeding time? Feeding time means for how long period you need to change your media, nutritional media. Like these all different things which is depend on proliferation time or doubling time, like how much food or how much nutrients it requires to double. You need to study that all these things about the cell lines once you purchase it. In non-proliferating culture, the medium will still need to be changed periodically, as the cells will still metabolize and some constituents of the medium will become exhausted and will degrade spontaneously. Like some are very spontaneously degradation means what? This is the very basic term. Some ions or cations are there, right? And these cations, they generally go and denature the protein, nutritional protein material, or very highly charged particles or ions which is present, and the cells are using as a food, or it is the like cofactor to any enzyme, and might be it get denatured or it gets stabilized because of these acidic components, which is so ultimately these nutrient compositions get degraded spontaneously, and that's what it is mean. Next one, you need to replace the media. There are 4 general observations are there. You need to study what is the pH. Once there is a drop in the pH, definitely there is a acidity increases, and you need to study, you need to change the media. Then second one is the cell proliferation or cell concentration. You need to study that. Once the confluence is there, you cannot hold on to the same culture flask. You need to subculture it immediately, and you need to divide these cells rapidly. What type of cell types is this? Depends on cell types. You need to prevent something which is going to degrade your media or cells. And the fourth one is morphological deterioration. Morphological deterioration means what? Suppose this is the cell flask. and these are our cell lines. My drawing is very bad, but chalo, just for a second just consider it is a good drawing for our study. This is the nucleus, right? These are cell lines, one above other. Okay, somehow they are cultured like that, confluence is there. Now take example, the cells which is present over here. I'll change the color so that I can show you. Cells which is present here, these cells. Now these cells immediately get media, right? It is exposed to external environment. But imagine the cells which is present over here, which is not exposed at all, because all three cells are above that cell, so it is not able to reach to nutrients so that it can absorb the food which is necessary to that cell. So it is just below these all three cell lines, so it is going to die immediately, and there is a deterioration, and might be there is a morphological deterioration, because the space is available to cells so that it can grow fibrous material to hold on, or it can show its own natural activity, whatever their shape and size is. But morphological, this cells is going to degrade. It's a smaller one because all the cells are pressing it at the bottom of the flask, and so ultimately there is a deterioration. So that concept is called it as morphological deterioration. I hope so you understood the concept. Subculturing, I already explained you in detail what is meant by subculturing, but just to take a revision, short revision. um, when the cell line is subcultured, the regrowth of the cells to a point ready for the next subculture usually follows the standard pattern. What is the standard pattern? Like, there is a graph. Take example, there is a graph, the cell, first cell. There is a primary cell is nurturing and it is getting nutrition, and then it is ready to divide. So at that time, that phase is called it as lag phase. That phase called lag phase. What is lag period? Lag period, after seeding, it is followed by the period of exponential growth called the log phase. When the cell's density is somewhat similar to the substrate with this equation, and reaches the level such that all of the available substrate is occupied, right? All of the substrate is occupied, or when the cell concentration exceeds the capacity of the medium and growth ceases or it greatly reduced. That's what we called it as uh, a lag phase. So this is the lag phase, and then the cell starts to grow, it starts to divide, and the growth medium from this point, now cells are not ready, it is growing, it is making mature themselves to regrow. Once it divides one after another, it reach up to that phase, and from this to this phase, the cultural aspect, this is called it as log or exponential phase, and that is also called it as log phase. So log phase is very much important. So whatever antibiotic testing in the immunology practicals or any immunological studies, whatever cells we observe, whatever cell lines or bacterial cell lines, even though at the time of antibiotic testing we observe, we generally take cells which is present at exponential phase or log phase. So remember this term. There are many times in GATE or NEET examination this question has been asked. At what stage of cells we generally test antibiotics or any other drug? So at the time of log phase, remember. So once they reach up to the confluency, now there is a stationary phase become. It is a plateau phase or stationary phase. There the division starts, and the cells try to degenerate themselves, or morphologically they deteriorate. So what is the best time for subculturing? Remember guys, this is at just near the position of exponential phase. This is the best timing to subculturing the cell line, before the confluency. Okay, not so much before the exponential phase or in between the phase. Here is the feeding time, because cell needs to grow and complete their a single lifespan or a single doubling time, and then there we need to change the culture. right? Next topic, next thing is how we subculture. I hope so we have studied these things, subculturing, what we do and how we do. I no need to repeat it again. um, okay, there is a one term that is meant by suspension culture. I need to tell you that suspension culture means there is a no addition of cell, the culture which is directly present in the flask and in a floating fashion. And such a type of culture we generally have is the hematopoietic cells or blood cells. We culture lymphocyte cells in the culture in the suspension fashion. That's what it is called, it as suspension culture. So there is no need of trypsin treatment, right? Because why do we need a trypsin? We need a trypsin to detach the cells which is attached to cell substrate, okay, to the bottom or substrate. So we need to break these contact here, and for that purpose we need a protease enzyme, and that is our trypsin. But it is not required over here, so it is quicker, of subculturing is quicker in the suspension culture, less traumatic for the cells because cells exposes less physical distraction or shaking, handling, chemical treatment gives cells a more morphological deterioration environment. So it get rid these suspension culture is actually face less traumatic effect while subculturing. Then scale up is easier. Scale up means growing the culture in multiple in the flask, so that scale up procedure is very good in suspension culture. This is the basic difference. Don't worry, I'm going to give you notes about this culture. I'm going to share my slideshow also. This is one of the steered tank bioreactor, steered culture, um, where we culture suspension culture industrially, not in laboratory. Okay, in laboratory we need just flask, we don't need any steered flask, right? Which is the stirrer is there, which is continuously moving with the on the magnetic stirrer, and it maintains its turbidity inside it. The next part we need to study is the insect cells, right? Insect cells, why we need insect cell lines? The insect cell lines, clearly it shows that in the insect cell lines insect cells are used. There has been considerable interest for for some time in the culture of insect cells for studies of pest control, right, or the environmental toxicology. Now here I'll just take an example. Recently there is an attack on, first of all, on the Pakistan of these, which species, you just tell me in the bottom of the comment, because where I'm going to share this lecture on the Google Classroom, you just mention that what was that species which is attacked first of all on the farm sector or farming of in the Pakistan, and then ultimately come along through Rajasthan, Gujarat and Madhya Pradesh, and it reached up to Maharashtra also, the upper part of Maharashtra. What is the pest, what is the name of that pest? So there are different pests, there are different insects which is comes in contact with our farmland and they attack. So ultimately we need to get rid of them, and for that purpose we need to grow the pesticides, we need to grow chemicals or any other biofertilizer agents to control that pest. So however, the greatest increase in the usage of insect cell culture has resulted from use of baculovirus gene cloning. Baculovirus is the type of virus which infects plant cells, right? Which infects plant. And so once we insert our gene of interest in the baculovirus and we allow that baculovirus to infect any plant, ultimately that plant will show us, or that plant will ultimately engulf that inserted genes with the function of baculovirus insertion methodology of gene in the plant, and plant will show us the various characters, right? That is one of the plant viruses we use for gene cloning. Baculovirus is often grow in SF9 cell lines. So baculovirus generally we produce in SF cell lines. So SF means what? That is the type of some sort of worm that is called it as army worm, and the name of that army worms is Podoptera frugiperda. This is the difficult word to pronounce, but it is the name of that Lepidoptera, or that is the insect one, arthropod species. It is the continuous cell lines. We infect that cell line first of all with this. In this method, SF9 cells are kept growing continuously in the magnetic stirrer culture flask, right, in the suspension culture. The flat bottom flask, obviously, with a magnetic stirrer. Then CO2 is not required because of it's an insect cell lines. They don't need CO2 because they don't need to, we don't need to maintain pH at all, because the cell lines of the insect shows some different properties. So I told you that the mammalian cell culture is completely different than that of the insect cell lines, and so insect cell lines shows different characteristic. They need a different requirement of their nutrient. They have a different genetic expression or phenotypic expression in the cell line. So with respect to that, we need to check or change our growth medium. And so for that purpose, the growth medium, we have this JRH Bioscience is one of the private company, pharmaceutical company or biological science company, which produces XL 400, and which contains 2 millimolar of L-glutamine supplemented with 5% of fecal bovine serum, then 5 ml of penicillin rhabdomycin as an antibiotic solution to, to avoid cross contamination of any bacterial species, right. The storage is at the 4 degrees centigrade in the dark, and always warm it in the room temperature before use. So the SF cell line, SF9 cell line, is the insect cell lines, where we need to study these insect cell lines with the different pest or different viral infections. So there is the basic application. Like once we choose the insect cell lines, we can study what is the effect of these pesticides on the insect, because ultimately we need to check whether that insect or whether that pest is dying or not, and what is the effect, whether it is breaking any protein which is secreted in the insects, or whether the any other metabolic process is disturbing because of our insecticide or pesticide. That all things we can study under these insect cell lines. I hope so you have understood. There is the routine maintenance that I have posted. It is from Ian Freshney book. I hope so you all know that book. I have already shared, but I'll again share that book. This is the typical maintenance of insect cell lines and then mammalian cell lines. So in the mammalian cell lines, I have told you that we generally use some sort of Hepes buffer system or different types of hormonal solutions that we generally used in mammary cell line or mammary epithelium cell line preparation, right. So this is the maintenance, and I hope so you have understood all our topic of the cell lines and the details about cell lines, right. So here for any queries you can post or you can question me on the Google Classroom, and I will post these notes with this current document. Okay, we'll meet on next lecture on cell characterization. Chalo, bye bye.